Welcome to Metalactome. Today we are going to discuss vitamin B1, which is also called thiamine or anti-Beri Beri. In this lecture, we will discuss the different sources of the thiamine, reactions of the thiamine, and what diseases that actually develop when there is a deficiency of the thiamine. So first of all, we will talk about the sources. So important sources, you will see the whole grain like wheat, barley and rice. But keep remember, these grain should be covered by the husk. So this point is very much important. Actually, the vitamin B1 is abundantly present in the husk. So next we will talk about the active form. Actually, thiamine exists in inactive form, and that form is the, here if you see, this is the thiamine. Thiamine exists in inactive form. It will convert into the active form by taking 2-phosphate group from the ATP. So, it will take the 2-phosphate group. Here is the ATP, and this ATP will give 2-phosphate group to the thiamine, and it will become AMP, adenosine monophosphate. Okay, now it will convert into the thiamine pyrophosphate. Now, this is the active form of the thiamine. This is the TPP, thiamine pyrophosphate. Now, keep remember that the thiamine pyrophosphate is the coenzyme. It works as a coenzyme. So this is the coenzyme form of the thiamine. Next we will discuss the difference between the alpha ketol and the alpha keto acid. Lot of students get confused between these two terminologies. So first of all we will discuss alpha ketol. That is also called alpha hydroxy ketone. So if you see that, it means two functional group is actually present, ketone and the alcohol. So if you see here, like in the ketone we see the alkyl group at one side, and here you will see hydroxy, and here is the R group. So if you see here that this is the functional group of the ketone. This is functional group of the ketone, and the carbon that is attached to the functional group is actually called the alpha carbon. And at the alpha carbon you will see the attachment of the hydroxide group. So that's why we call alpha ketol. Keto means something related to the ketone, and OL, that is something related to the alcohol, and alcohol having functional group OH. So that's why we call it alpha ketone, or you can also say alpha hydroxy ketone. Next, if you see alpha keto acid, it means two functional group will be present. In that case you will see acid carboxylic group and the ketone group. So if you see here, like this is the carboxylic group COOH. Now here is the alpha carbon, and at the alpha carbon you will see the presence of the ketone. So if you see here, like this is the carboxylic acid group, and here you will see this is the alpha carbon, and at the alpha carbon you will see the ketone group. So now that's why it is called the alpha keto acid. So now hope so you got the point between the basic difference between the alpha ketol and the alpha keto acid. Next we will discuss the reaction, how thiamine pyrophosphate involved or act as a coenzyme in different reactions. So as we know that when we take food, we actually take the glucose in our body, and that glucose is break down in our body through the respiratory process. So in the respiratory process, you will see the three main important processes. So if you see here, first process is the glycolysis, second process is the Krebs cycle, and last process is the electron transport chain. But our main focus is on these two processes, glycolysis and Krebs cycle. Okay, now two most important reaction, formation and the degradation of the alpha ketol, which we have just discussed. Alpha ketol having ketone and the alcohol functional group, and oxidative decarboxylation of alpha keto acid. Now this is the other thing. Now first of all we will discuss the how alpha ketol is formed. Actually alpha ketol is actually a group, functional group, combination of functional group that are actually present in different compounds. We will see it later. Here if you see, first of all in the glycolysis we see that the glucose is converted into the pyruvate. So if you see here that the glucose is converted into the pyruvate, and in this process this will go through the different processes like glucose converted into the glucose 6-phosphate and lot of different processes, and ultimately you will see the formation of the pyruvate. But in some situation glucose 6-phosphate will go to the hexose monophosphate shunt, and you will see that glucose 6-phosphate will form or will convert into the ribulose 5-phosphate with the help of the shunt pathway. So, but keep remember, in the conversion from the glucose 6-phosphate to the ribulose 5-phosphate, you will see the lot of different compounds like the 6-phosphogluconolactone, 6-phosphogluconate, and ultimately you will see the ribulose 5-phosphate. Okay, now if you see that the ribulose 5-phosphate will convert into two different compounds. First compound is the ribose 5-phosphate and second compound is the xylulose 5-phosphate. These are basically the two different compounds. And keep remember, these compounds contain 5 carbon and this compound also contain the 5 carbon. Okay, now this is the 5 carbon compound, this is the 5 carbon compound, and after that you will see ribose 5-phosphate will convert into the, here is the glyceraldehyde 3-phosphate, and the xylulose 5-phosphate will convert into the pseudoheptulase 7-phosphate. But there is a change, you will see the changes. This is actually the 3 carbon compound and this is actually the 7 carbon compound. But why? It is xylulose 5 carbon. Then how it will become 7 carbon? Because 2 carbon from the glyceraldehyde goes towards the pseudoheptulase 7-phosphate. Now you will see 5 carbon, 3 carbon goes toward glyceraldehyde and 2 carbon will goes toward the pseudoheptulate. So 5 plus 2 will become the 7 carbon. So this is the pseudoheptulate is the 7 carbon compound. Okay. Now in this conversion from the ribulose 5-phosphate to glyceraldehyde 3-phosphate and the xylulose 5-phosphate to pseudoheptulase 7-phosphate, you will see the enzyme, and that enzyme is very much important, and this enzyme is actually the ketolase, this enzyme. Now this enzyme actually involved in the conversion of all these processes, and this enzyme required the coenzyme, cofactor, and that coenzyme is actually the TPP, thiamine pyrophosphate. So this enzyme will take the TPP as a coenzyme. So now this point is very much important. Okay, why we call the formation and degradation of alpha ketol? It means there is a compound having alpha ketol functional group, and that compound is actually the pseudoheptulase, also contain the alpha ketol. Xylulose also contain the alpha ketol functional group. Now if you see from the ribulose 5-phosphate, you will see the formation of xylulose. It means formation of the alpha ketol, and it will after that you will see it will degrade into the pseudoheptulase. So it means that the alpha ketol is form and degrade continuously, and ultimately you will see the pseudoheptulase also contain the alpha ketol. It will further degrade into the erythrose 4-phosphate. That's the other process. Next, if we see that the oxidative decarboxylation, in this case we see that alpha keto acid. So keep remember that the pyruvic acid pyruvic acid and the alpha keto glutarate, that is actually the part of the Krebs cycle, contains the alpha keto acid. If I write the structures of the pyruvic acid, then you can easily understand. Actually, pyruvic acid having carboxylic function group. Along with that, you will see the ketone group. This is the carboxylic acid and this is the ketone group. Now this is the structure of the pyruvic acid. That's why we call it alpha keto acid. Just like alpha keto glutarate also contain the alpha keto acid group. Now pyruvate convert into the acetyl CoA with the help of the pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex. This is actually a complex. This complex contain different enzymes like E1 enzyme, E1, E2, E3. But our main focus is on the E1. E1 is actually the pyruvate decarboxylase enzyme. So pyruvate decarboxylase enzyme actually involved in the conversion of pyruvate to acetyl CoA. Lot of different enzymes, even though lot of different enzymes involved, but our main focus is the pyruvate decarboxylate. If we focus on the word oxidative decarboxylation, oxidative, that is something related to the oxidation. In this process, you will see oxidation means removal of hydrogen. In this process, you will see the hydrogen removed from the pyruvate. So NAD positive will convert into the NADH. NADH, H comes from the pyruvate. So this is the oxidation. Now decarboxylation, removal of the carbon dioxide. In this process, you also see the removal of the carbon dioxide. Now this process is the oxidative decarboxylation process in the conversion of the pyruvate to the acetyl CoA. Next, if we move towards the Krebs cycle, you will also see from the alpha keto glutarate to succinyl conversion, same process, you will see the formation, oxidation, NAD2, NADH, and you also see the removal of the carbon dioxide, that is decarboxylation. Now, in this process, you also see the enzyme and you also see the keto glutarate dehydrogenase complex. Now this complex contain also different enzyme, but the important enzyme is the glutarate decarboxylase, and that enzyme is actually involved in the conversion of the alpha keto glutarate to the succinyl CoA. And you see that this enzyme can require coenzyme or cofactor, and that is the TPP, and this enzyme also require the TPP. Now overall, if we summarize that the pyruvate decarboxylase require the TPP as a coenzyme. Alpha keto glutarate decarboxylase also require the TPP as the coenzyme. So if I say that when there is a deficiency of TPP, then the whole process, Krebs cycle or respiratory cycle, becomes stopped. And overall, as we know that the respiratory process happened, main purpose of the respiration is the production of the energy in the form of ATP. Then ATP will reduce and the cellular function will become impaired. So if I say that if the ATP reduce due to the when there is a deficiency of TTP, all process become reduced and reduction of ATP and the cellular function become impaired. Next is the, if I block this process, then the level of the pyruvate become increased. In this case, so it's mean that the pyruvate will start accumulating in our brain and cause of different diseases. So this is the deficiency of the thiamine. Next we talk about the what diseases that actually develop in thiamine deficiency. So first one is the Beri Beri. In the Beri Beri you will see the three types: dry, wet and infantile. So you will see the dry Beri Beri, wet Beri Beri and infantile Beri Beri. Okay. Dry and wet will be adult related and infantile, that is the related to the infants. In the dry Beri Beri, you will see the most important thing is the polyneuritis. So this is the polyneuritis. This is actually the disorders of the peripheral nerves. This is actually you will see in the dry Beri Beri. And in this case, you will see the most important muscles will be affected in dry Beri Beri, and that is the calf muscles. Calf muscles is mostly affected by the dry Beri Beri. And you see the most important symptom, and that symptom is the burning sensation, burning feet syndrome. That's also see burning feet syndrome. You can also see in this case in the dry Beri Beri. So these two important things. Next is the wet Beri Beri. Wet Beri Beri, in the wet Beri Beri you will see the edema. So edema, and you can remember in the wet you will see the presence of E alphabet, and that E alphabet will remind you that it is something related to the edema. Why edema happens in this case? Due to the dilated cardiomyopathy. So due to the dilated cardiomyopathy, in this case you will see the heart muscles become weak and dilate. So due to the heart weakening of the heart muscles, you will see the heart cannot pump properly, and in this case you will see the edema. But keep remember that in the most heart diseases you will see the edema, and edema is also the sign of the heart failure. Okay, in fertile we will talk about that is something related to the cardiovascular abnormality you will see the infertile, and that is mostly happens in the first year of life. Okay, now if we talk about the second disease is the Wernicke-Korsakoff syndrome. We divide Wernicke-Korsakoff syndrome. It is actually happens in a different situation like alcoholism, and when there is malabsorption of the vitamin due to gastric surgery. So we divide Wernicke-Korsakoff syndrome into two. First one is the Wernicke encephalopathy, and second one is the Korsakoff and psychosis. In the Wernicke encephalopathy, you will see the damage of the part of the brain. And what part actually damaged in this case? You will see the thalamus and hypothalamus. These two parts of the brain mostly affected in the encephalopathy, Wernicke. And symptoms you will see, like in this case you will see different symptoms like ophthalmoplegia, and you will see nystagmus, diplopia. These are actually the diplopia, ophthalmoplegia. That is actually the weakening of the muscles of the eye. So these are basically the different symptoms in the Wernicke encephalopathy. Psychosis, Korsakoff psychosis, you will see the damage of the part of the brain that is related to the memory. Those part of the brain will become damaged. That is something related to the memory generation. So in this case, you will see the hallucination, both visual and auditory. Hallucination means making perception that someone is walking outside or someone is going upstairs, but in reality this is not the case. So this is actually the hallucination. Visual, that is related to the watching perception, and the auditory, that is related to the sound perception. So hallucination, and in this case you will see, you can also see the memory loss. So memory loss, and you can also see the confabulation, making different stories, like you can consider like hospital as a house. So this is actually the different story, but in reality it is not the case. So this is all about the vitamin B1, which is also called the thiamine. If you have any question, then you may ask in the comment section. Thank you.